much wrap up chapter 4. Then we'll move into chapter 5. The important part about chapter 5 is it's called Extensions and Modifications of Mendelian Genetics. What you're really going to see here is examples where the phenotype would look at first glance like it was actually some sort of exception to Mendel's laws for genetic inheritance. However, what you're really looking at, and the important point to think about, is that the phenotype is the result of how the genes are expressed, and in some cases how the products of those genes interact with each other. But in all cases, the actual inheritance of the genes will still follow Mendel's laws as he laid them out. It's these complications of how the genes interact that Mendel had a very nice, clean, maybe he was a little lucky system, and if any of the events that we're talking about in this chapter 5 had been present, it would have thrown off the ratios that he saw extensively. He may have observed in certain phenotypes related to what we're going to talk about and just chose to ignore them, to keep his experimental model as simple and straightforward as possible. So we're going to get most of the way through chapter 5. I doubt seriously if we're going to finish it, but what that means is that we're going to have probably about 90% of the material for exam 1 finished today. So we'll finish this up next Monday. You'll have a full week. We're going to continue on with the lectures, but you're going to have a full week before you're actually going to be tested on that material. At the end of the lecture on next Wednesday, then I'll stop about 20 minutes early, and at that time I'll just sort of open up the floor for discussion of any questions and such that you might have. So as much as I can, I'll give you this almost a week advance in preparation for the exam. Obviously, I can't do that at the end of the semester, okay? But it usually works out pretty well. All right. So we're towards the end of chapter 4. We're talking about sex-linked inheritance. The key to this being that in males you have this Y chromosome that only shares a very limited amount of sequence homology with the X chromosome. Therefore, virtually all the genes on the X chromosome, or let's put it this way, virtually none of the genes on the X chromosome are present on the Y. So in males, they only have one copy of each of those genes. If there is a recessive mutation, they're going to see that phenotype because there's no wild-type allele to show dominance with it. And therefore, we use that term hemizygous. Whereas in females, it's more like an autosome situation. You have the two X chromosomes. So if they're heterozygotes and it's a recessive mutation, you're going to see the wild-type phenotype. Otherwise, the segregation of the chromosomes, they go exactly as the autosomes. They go through meiosis in exactly the same way. They follow all of Mendel's laws for gene segregation. Yes?
You won't see a wild type with males?
Not if you have the recessive mutation, because males are effectively hemizygous. But this sets up a problem, and this is where we ended at last time, is this idea of dosage compensation. If females have 2 copies of the genes on the X chromosome, and males only have 1, then there's a gene imbalance. At a cellular level, the cells really want similar levels of gene expression off of those genes. And so between mammals and Drosophila, this is another one of those contrasting situations where the outcome is the same, but the problem solved in 2 different ways. Okay. In flies, it's a very straightforward approach. You have 2 X chromosomes in females, you have 1 X chromosome in males. The genes on the X chromosome in males are hyperactivated. Simply means they're transcribed at twice the rate as they are in females. So those genes on the X chromosome in males in flies, in Drosophila, are working twice as hard, producing twice as much mRNA per gene. So at the end, you have similar levels of gene expression in both males and females. All right. But there is a regulatory mechanism, we won't go into it, that only hyperactivates the genes on the X chromosome in males. Would there be a problem if there was a mutation that hyperactivated the genes on the autosomes? Yeah, then you'd be back to that dosage imbalance again. Okay. Mammals have taken a different approach. For mammals, the idea is inactivate all of the X chromosomes except for one. So we already talked about how it's essential that you have one X chromosome. There are no known examples of a viable individual who had no X chromosomes. Okay. So what happens is in the normal situation for females, you got two X chromosomes. One of those X chromosomes is going to become inactivated. It's basically going to condense. We'll talk about that a little bit later. And transcription off of that chromosome is essentially zero. This condensation, this sort of collapsing of what we call heterochromatin, formation of heterochromatin, now gives you can actually see this in the interphase nuclei as a bar body. So this is, if you use a dye that stains the DNA, then what you see is this very darkly staining body which represents this bar body. That's the inactivated X chromosome. Okay. But it does set up an interesting situation. So males are genetically hemizygous for the X chromosome. They only have one copy of the X. But females are functionally hemizygous at the cellular level because they only have one functional X chromosome in the cell. If the female is heterozygous for a particular gene, has two different alleles, then she's really a genetic mosaic. Depending on which X chromosome gets inactivated, you're expressing different alleles. All right? So how does this come about? As you go through development, at some point in development, this differs with different tissues, but a decision is made, a genetic decision, to inactivate an X chromosome. As far as we can tell, that decision to inactivate that X chromosome is random. However, once that X chromosome is inactivated, let's say it's the maternal X chromosome, all of the cells derived from that cell all have the same X chromosome inactivated. But in another cell, it may be the paternal X chromosome got inactivated, and then all those cells derived from there all have the same chromosome inactivated. So you can have patches of cells that are expressing one allele and patches of cells in another part of the organism expressing a different allele. Okay. And so here's a pretty colorful example, this so-called tortoiseshell. So what you have here is a gene on the X chromosome that's involved in fur color. Females who are homozygous wild type have black fur. If they're homozygous for this O allele, they'll produce orange fur. But heterozygotes give you this patchwork. So what it's saying is, in this part of the fur here that's orange, the X chromosome, which X chromosome was inactivated? The one with the wild type allele. And over here in the black fur, it was the opposite. In this case, the X chromosome with the O allele, the recessive mutation, was inactivated. So you get this patchwork because it's really a genetic mosaic. All right. Now, yes? No. No, it's actually, we'll talk about that in a few minutes. No, it's the fact that genetically, the genes being expressed in this tissue are different than the ones here, a completely different allele. Because you have two possible options, but you take one of them out of the game. And because if there was some molecular mechanism that says wild type, you always inactivate the X chromosome with the non-wild type allele, would you see this effect? No, it's because this inactivation appears to be random. Would you see this effect if the individual is homozygous for that particular gene? No, if it's the same allele, it's not. You only see it when they're heterozygous because of this random inactivation. All right? Now, males should either be black or orange. Does that make sense? Okay. How do you end up with a tortoiseshell male? Yeah, the answer's right up there. You could have an XXY individual, which would be male, but just because it's male, it still will have the bar bodies. You'll still see the random inactivation of one of the X's. Good question?
So if they have more than one X chromosome, then they'll have themselves?
Yes. So I thought maybe the other question, because I only thought about it, was what if this was a triple X female, and there were 3 different alleles? Maybe one for black fur, one for orange, and one for white. What would you see? You see all 3 colors because of random inactivation. But I think the answer to your question is, regardless how many X chromosomes are present, you'll have bar bodies equal to that total number minus 1. It'll just be 1 inactive X. All of the others will form bar bodies. Right, males would have an aberrant male in the sense that it's XXY has to do the same thing. It has to do this gene dosage balancing. Okay. All right. So to just begin to touch a little bit on molecular biology, just to sort of touch on the molecular mechanism of how this works, and we'll just sort of glance over it. We're going to come back to some of these mechanisms, but just to kind of introduce it. The key gene that's involved in X chromosome inactivation is called Zeiss, for X inactivation specific transcript. What's a transcript? Are you familiar with that term yet? It's another name for RNA. It's from transcription. So many times mRNAs, we simply refer to them as transcripts. So if you see, such and such transcript, it's referring to the RNA from it, usually an mRNA. All right. What's the normal product of a gene? The final product is normally what? A protein. In this case, the final product is an RNA, not a protein. It stops at the level of being an RNA, and it's a DNA binding RNA. So there's a specific site on the X chromosome that is to be inactivated, and this Zeiss transcript binds to that DNA site. That's the molecular trigger that sets off a series of biochemical changes. What we're trying to do is condense the chromosome. So we've already shown where we went through meiosis and you saw the chromatin begins to condense, and ultimately you get these highly condensed metaphase chromosomes. That very highly condensed chromatin, called heterochromatin, is usually what you find in areas of the genome that are not being transcribed. So the way to inactivate this X chromosome is basically fold it all up, crush it all down, get it all compact, and make it transcriptionally inactive. So that's what you're really seeing in the bar body, is this condensation of this X chromosome that's being initiated by the binding of the Zeiss RNA to its target sequence on the chromosome. There's a lot of biochemical changes that go with this. We're not going to go into this in detail. We'll come back later. But for instance, we'll talk about acetyl groups that are on histone proteins. How tightly those histone proteins bind to the DNA depends on how they're charged. The acetylated histones can't bind as well. Therefore, the chromatin is a little looser. If you remove the acetyl group, then the histones can bind tighter. The chromatin gets a little more condensed. There's some methylation of the histones. There's some other changes. But again, the key here is the binding of the Zeiss transcript sets off this series of biochemical changes that results in this X chromosome condensing and forming this bar body. Okay. Now the other question's got to be, well, what keeps it from inactivating X chromosomes? There's a putative inhibitor of Zeiss. That one's not really as well characterized. So this is where we are with a lot of these complex cellular events. We're beginning to understand the molecular mechanisms, but in many cases we don't have the whole story yet. There's still some black boxes out there, which at least years ago my cell biology professor used to point out that since there's unanswered questions here, then you all, me when I was a student sitting out there, have an opportunity to have a career. If we already have all the answers, then we don't have to do any further research. We're not even close to having all the answers. Okay. Now there are a few cases, aberrant cases, where it looks like the X chromosome, this process can get reversed and the X chromosome is reactivated. And I only point that out is it's looking now that there's some evidence that in certain cancers, in fact, such as in some breast and ovarian cancers, it looks like what was the inactive bar body has become a reactivated X chromosome. Maybe this dosage compensation now, overexpressing the genes, is part of what's leading to this disease process. Okay. Later, at the end of the course, we're going to barely have time to talk about it, but the so-called oncogenes, the cancer genes, are actually normal genes. The normal genes called proto-oncogenes, Steve Bishop got the Nobel Prize for finding that, are genes that are expressed at the wrong time, in the wrong tissue, in the wrong amounts, and then a normal gene being expressed in this aberrant way actually leads to the cancer process. So, okay. Yes. Okay. So as I've kept stressing is there are a number of things about Mendel's experiments that really worked in his favor and allowed him to come to the correct conclusions that he did about how genes are transmitted from one generation to the next. However, once his work was sort of rediscovered, about 30 years after he passed away, and more experiments were being done, especially in Morgan's lab with Drosophila and such. We already talked about the white-eye mutation. There are lots of examples that started coming out that said, at least based on the phenotypes, is this doesn't look like it follows Mendel's laws. Again, the key to this, and here's probably, I think, the most important point, is the segregation of the genes in each case follow Mendel's laws just as we've been talking about them. The difference is we're looking at the phenotype, which is the result of how those genes are expressed, and in some cases how the products of those genes interact with each other. So it's not the genes per se when you see the phenotype, but the end result is really what is the product of that gene and how is the product of that gene, usually a protein, how is it interacting with other proteins in the cell to give you the phenotype. Okay. All right. Now, this particular one, I'm going to start out with this first discussion. I didn't quite notice until this morning that he had moved this discussion a couple of slides down. It's still the same. I decided to just leave it where it was. So this is actually figure 5.4 in the new version of the text, just to keep it consistent with your notes. We're covering the same material, and I couldn't see any reason why we had to present it in different ways. Okay. Anybody recognize, what are these? Okay, see, you can answer these questions. All right. Okay, and how are they different? They're in different colors. Okay. So you have this yellow mouse. All right. So it looks like a typical Mendelian cross. You got a heterozygote. Let's assume that one gene, there's just a gene we'll call Y, is responsible for this yellow fur, and you cross a male and a female. They're both yellow. So you have these two heterozygotes. And what you find is you still get the same segregation. 1/4 are homozygous big Y, 1/2 are heterozygotes, and 1/4 are small y. However, the quarter, what you really find when you start looking through the litter and you start counting animals, is you get 2/3 yellow and 1/3 white, or non-yellow. Because you never see these animals die in utero. Okay, so this is what we call a recessive. This is what we call a lethal allele. It's a gene that is absolutely critical to development. If it's mutated and non-functional, then when it's homozygous, it's lethal, and the animal never develops. And so the first time you get a hint of this was when you're looking through the progeny and you're not getting the ratios that you expected. Okay.
So what you're saying is basically the recessive gene is lethal?
It gets more complicated. Okay. So as I said, lethal mutations are mutations in genes that are absolutely essential for development. And so when they're not functioning properly, the homozygous recessive mutations, then it's lethal. So, are all lethal alleles recessive, or could you have a dominant lethal mutation? Okay? And just as a side story, because I do this, when I was in grad school, played on a co-ed softball team, which everybody wanted to play against us because it was pretty much a sure win, but we had a lot of fun. And we named ourselves the dominant lethals, but it turns out if you're not a geneticist, that doesn't intimidate anybody because they have no idea what you're talking about. Okay? So could you have a dominant lethal mutation? A dominant lethal mutation would say what? That the heterozygote lives or dies? Dies. So you really couldn't have a dominant lethal mutation. I'll make the argument that mutations like Huntington's disease, which is caused by a dominant mutation, is a dominant lethal. Okay? But why does it survive? Exactly. So these recessive lethal mutations, so the answer was they survive past the point, the typical reproductive age. So these recessive lethal mutations, the ones we normally are always thinking about, are going to cause the animal to die before they reach reproductive age. So you couldn't have a dominant lethal, it could never carry through to the next generation. Okay? So the vast majority of the typical lethal mutations that we're talking about are recessive. Okay, but here's where it gets a little more complicated. We got our definition of dominant and recessiveness from Mendel's very simple experiments. All right? So we've used a big Y to indicate the dominant allele. The big Y, isn't that, we're saying homozygous dominant, doesn't that make it a dominant lethal? There's really two phenotypes here. The Y is really dominant for hair color because when you have a heterozygote, it has yellow hair. But it's recessive with respect to lethality because the heterozygote is viable. So it's one of these complications where how do we designate this? Do we use uppercase or lowercase? Because with one phenotype, it's dominant. With the other phenotype, it's recessive. Okay? Just starting to introduce some of the complications that come in and why we have to keep modifying what we mean by some of these terms like gene, dominant, recessive, homozygous, heterozygous, as we understand them at a more detailed level. Okay, confusing enough? Yeah, we're getting there. Okay. Same sort of thing, this goes to your question about incomplete dominance. In Mendel's case, every trait that he looked at, when he had the true breeding parent had one phenotype and the other parent had another phenotype, when he crossed them together and created the heterozygote, only one phenotype came through. And that's what he called dominant. All right? And he didn't see that second phenotype, he didn't see that recessive phenotype until the next generation. But that's because the heterozygote was indistinguishable from the homozygous wild type. But incomplete dominance occurs when, in fact, the heterozygote has a phenotype that's distinct from both homozygous dominant and homozygous recessive, but lies somewhere in between. All right. So a very common example of this is pigmentations. It's kind of like mixing paint. So up here, if we had two true breeding, this was a Mendelian experiment. We have one plant that has all red flowers, and we have another plant that has all white flowers. With that information alone, do you know which one's dominant? Actually, you don't. You'll make an assumption that the red one probably is, but not necessarily. But now when you make the cross, and this is what Mendel was looking at. He had these two traits. He didn't know which, so when he made the cross, if he got red flowers, he called the A1 allele dominant. If he got white flowers, he called the A2 allele dominant. But in many cases, the heterozygote is sort of this pink, something in between. So it's this incomplete dominance. So you have incomplete dominance when you can distinguish the heterozygote from both the homozygous dominant, homozygous recessive, and that phenotype lies somewhere within the boundary between the two. So you got red flowers, white flowers. The heterozygote is somewhere in this realm of pink. Okay, that was a pretty straightforward. Any question? Okay, so here's it, just writing it out for you, what we just talked about. All right. So the key here is that this dominance really affects the phenotype. What we call dominance affects the phenotype. You don't inherit that. The genes still segregate, just as Mendel said. It's the way those genes are being expressed. Okay. Codominance is just an extension in a situation where we don't have a clear dominance. We can't really argue that one allele is more important or is completely dominant to the other. Some of the major examples here, the easiest ones are things like antigens, proteins that are on the surface of cells. So I'll show you a couple of examples from blood cells. So there is a set of antigens called the MN blood antigens. They're not as immunoreactive as the ABO and RH, so you don't hear about them as much, but they're on the surface of the red blood cells. There's one gene. The M allele makes an antigen called M, just a protein on the surface of the cell. N codes for a variant of that protein called N. So the homozygotes only have the M antigen. Homozygotes for the M allele only have the M antigen. Homozygotes for the N antigen only have the N antigen. And the heterozygote expresses both. Which one's dominant? Is one any more dominant? So it's codominance when we really can't assign any sort of a dominance or recessiveness to this phenotype. Yes?
So, for example, for the flowers, A1 and A2 are expressed in, were both expressed in the flowers, like pink. So
right.
Is that in the codominant as well?
No. No, because clearly here, let's assume this is what you saw when you made the heterozygote. Clearly A1 is dominant to the A2 allele.
Yeah, I'm talking about the pink flower.
Right. So down here, but the reason this isn't codominance, but it's incomplete dominance, is because we can clearly say that the red is dominant to white. And it's incomplete, not quite fully dominant, because it's somewhere in between. Yes?
So flowers with spotted white and red would be codominant, showing both of them?
I would call it codominant, yeah. It's within the range of the two extremes. If they were blue, then it becomes another situation. But as long as they're within the... And so these pigmentations typically fall into this kind of incomplete dominance. There's another question?
Yeah, that was the same.
Oh, okay. So the difference here is with the codominance is you really can't establish that M is more important, is dominant to N. It's really just the presence of the proteins on the surface. So we simply call them codominant. Okay, that's what we did. Here's an example where we have to define what we mean by dominant and recessive. Okay, Mendel made it very simple. Whatever trait I see at the F1 generation, I'm going to call it dominant. All right. We look at something like cystic fibrosis. We know this is caused by a mutation in a single gene. The gene it's an autosomal gene. It's on chromosome 7. The phenotype, the clinical phenotype, is individuals with cystic fibrosis produce large amounts of mucus. This clogs up the lungs and the ducts and the pancreas and such. The gene is actually called CFTR for the cystic fibrosis transmembrane conductance regulator. Basically, it's a cell membrane protein that regulates the flow of chloride ions in and out of the cells. So all of this devastating results from cystic fibrosis is just from an inability to properly move chloride ions across the cell membrane. It's a recessive mutation because individuals who have the clinical symptoms are always homozygous for the recessive allele. Heterozygotes are normal. They don't have the clinical symptoms. However, heterozygotes have one functional allele and one non-functional allele. But they're both expressed, so they're making two different proteins. One's functional, one's non-functional. All right? So really it's codominance at the molecular level. But the heterozygotes don't develop the disease. So what we mean by whether we're going to call it dominance, incomplete dominance, codominance, we really have to define what we mean by it for that particular example. So as we go into the molecular levels, we'll see more of these examples where we can find the heterozygote if we look at the molecular level. If we just look at the gross phenotype, we won't be able to find it. And therefore, well, we call this dominance. So it's this constant adjusting of this definition. Okay, to sort of summarize, and here's an important point. Dominance is really, what we're looking at is the interaction between different alleles at the same gene, at the same locus. Because in a few minutes, we'll be talking about interactions between genes at two different loci. So these are allelic interactions. All right. Again, reiterating the point that it doesn't affect how the genes are inherited. The phenotype didn't run into, or may have and just chose to ignore, is a term we call penetrance. In Mendel's experiments, every plant that had, let's say, the wild type allele for the wild type seed color always had that phenotype. There was a 100% correlation between the phenotype and the genotype. That doesn't always happen, especially in a lot of disease situations. What we mean by penetrance is what percent of the individuals have the phenotype who have the genotype. Right? 100% penetrance means every individual that has this specific genotype, we can predict will have this phenotype. But that doesn't always happen. So here's an example that comes out of the text. Polydactyly is this having extra fingers and toes. And in this case, it's usually caused by a dominant allele. But what you find is you can find individuals who have the normal number of fingers and toes, yet their children inherit this condition. All right? So you immediately, the first thing you suspect is the father, right? Okay. Ruling that out, okay, what you find is this gene for polydactyly is not fully penetrant. There are individuals who will have the dominant allele for this particular gene but will not show the trait. All right? So penetrance is the percentage of individuals having a particular genotype who also express the expected phenotype. In all the experiments that Mendel did, penetrance was what? It was 100%. If any of those traits had been 50%, 60, 70% penetrant, he would have seen what? When he started looking at all his careful little ratios, it would have been very different than what he was seeing in the other experiments. Okay. Related to this is also what we call expressivity. The phenotype, even if the individual has the phenotype may even vary. So with polydactyly, what you can find is they have the same dominant mutation, but the severity of the condition can vary from fully functional digits to just little bits of skin. So it's variable expressivity. So just because they have a specific genotype doesn't mean you can predict the phenotype with complete accuracy. They may not have the condition at all, or even individuals with the same genotype may have variations in how severe the condition is. So this is the expressivity, how severe is that phenotype, even though these two individuals have the same genotype. The reason we see that is because these genes don't function in isolation. So the gene that's encoded, this gene with the dominant mutation, the protein product of that, interacts with other proteins, interacts with environmental signals, and therefore the outcome depends on these other factors. So if, for instance, I don't know enough about the disease, but let's say this protein has to interact with some other particular protein that happens to be absent. If it's not there, then you don't have the condition. That might be enough to explain this less than 100% penetrance. As I said, you see this a lot of times in disease situations, especially with cancers. That's why things like BRCA1 and 2 mutations, although it may dramatically increase a woman's chances of developing breast cancer, it's not absolute because there are other factors, especially environmental factors, that come into play. All right. Next one is multiple alleles. Again, Mendel kept it simple. He dealt with 2 alleles for every trait he was looking at. What was probably the wild type of what he called the dominant and a recessive mutation. Well, we've talked about this before. Are there, for any given gene, are there only 2 alleles out there in the real world? Well, you could have any range of multiple alleles, even though any individual can only have a maximum of 2 different alleles. So what we're looking at here is give you an example of a situation where there are multiple alleles and how you get different phenotypes, but again, watching that the segregation and transmission of the genes still follows the Mendelian laws. Okay. So this is the example that from the book are duck feathers patterns. So in this case, to keep it fairly simple, there's just 3 alleles we're going to deal with. The wild type for mallard is an uppercase M. A pattern called restricted, which I'm not a duck hunter, I don't know what really that is. So the R allele, so it's just, keep in mind, it's one gene. Gene M, there's 3 different alleles. The wild type allele, the R allele, which gives this restricted pattern, and small d for dusky. The key to this is the order of dominance. The restricted is dominant to the other 2 alleles, and the dusky is recessive to all of the alleles. All right. So there's 6 different genotypes that are possible. Should you memorize this equation? You can if you want to, but don't worry about it. Here's the key point. If you have this heterozygote, because R is dominant to the dusky, it has the restricted phenotype. This has got the mallard phenotype because big M, the uppercase M allele, is dominant to dusky. Okay. Now you get, here's the gametes. Any deviation from Mendel's laws there? All right. You go through fertilization. You get the same distribution of alleles that you would get using your Punnett. Could you do this with a Punnett square? Sure. You'd have these 2 alleles along the top, these 2 on the side. You put them together, you get all of these combinations. So this all follows Mendelian segregation. However, it's just the dominance of the different phenotypes. So this one's restricted because R is dominant to mallard. This one's also restricted because it's dominant to dusky. This one's mallard because it's dominant to dusky, and this one is got the fully recessive phenotype. So it's just multiple. It's the same concept. It's just multiple alleles are involved, and you just have to know the dominance order of the different alleles. All right. Here's one that may be a little more familiar: the ABO blood types in mammals. Okay, it's another multiple allele system. Again, we're just dealing with one gene. The I gene codes for the ABO antigens. So the A allele codes for the A antigen. This is a protein on the surface. B codes for the B antigen. And small I makes a non-functional protein, so there's no antigen present. The dominance pattern, so A would be dominant to small I. B would be dominant to small I. But A is codominant to B. All right, they're of equal importance, and one particular phenotype doesn't dominate over the other. All right. So, as you probably should already be aware, your immune system makes antibodies against the foreign antigens. Okay, so this is why it has to be considered in blood transfusions. So, blood type A will produce antibodies against B. Blood type B will produce antibodies against A. AB doesn't make any antibodies. and O makes antibodies against both A and B. Right. So what you want to see here is, here's the phenotype of the blood type, and you have two possible genotypes. For instance, I can give you type A blood. You can either be homozygous for A, or you can be a heterozygote for A in the small I allele. Does that make sense? So this heterozygote is only making the A antigen. I encodes the non-functional protein, so you have no antigen. Similar situation with B. A, B, there's only one genotype possible. Okay? And O has only one genotype. And then this just shows the typical agglutination reactions and why O is the universal donor and AB is the universal recipient. But again, it's a multiple allele system showing both codominance and complete dominance. All right. So any questions on what we've talked about up to this point, which have all been modifications of Mendel's observations, but they only involved one gene? Now we're going to start a discussion where we're seeing a phenotype that's actually the result of the interaction of two different genes. All right. So you got to keep in mind that what you really have in most cases is a protein product, usually an enzyme. What we're going to see here is these proteins interact with each other in some way to give you the new phenotype. Mendel didn't see any. This is another one of the simplifications. Okay? If any of the genes that he was looking at interacted with each other in any way, they would have seen some new phenotypes. But they all functioned independently, and so he never saw any of these type of gene interactions. Okay? So in Mendel's dihybrid experiments, because one set of alleles, the expression of those alleles had no impact on the expression of other ones, this is where he saw this 9:3 to 3:1 phenotypic ratio. If you see variations of that, it begins to tell you right away you're probably looking at some gene interactions. So that's where we're going with this, is to show you that you can get altered ratios, but they fundamentally always come back to this 9:3 to 3:1, which tells you you're looking at the interactions between two different genes. Okay. So what you're going to have is independent assortment of the genes. That is, think of them as the genes are on different chromosomes, but the products of the genes are going to interact with each other. Okay. So, and this can involve lots of loci. You can see this could get very complicated. We're just going to limit ourselves to two genes. Okay. So here's one example from the text. So you're looking at coloration of peppers. And there's 4 different colors: red, peach, orange, and cream. If you take homozygous red peppers and cross those with homozygous cream-colored peppers, all the F1 are red peppers. So what does that look like? What's your simplest interpretation? Right, there's probably one gene. It's a simple Mendelian cross. There's one gene that controls the color. So now you do a self-cross of the F1, and you get 9 peach, 3 orange, 3 yellow, and 1 cream. Now what does it look like? That there's it's a dihybrid cross. There's probably 2 genes involved. So the first set of data says one monohybrid cross, second says a dihybrid cross. What's happening here is the coloration of these peppers is really dependent on 2 different gene products. And they're going to interact with each other to affect the color. So you've really got 2 different genes, 2 different loci. So the dominant big R allele at the R gene, that produces a red pigment. The recessive allele, small r, makes no pigment at all. But there's also a gene involved in chlorophyll stability. So the dominant allele, big C, leads to a breakdown of the chlorophyll. That's going to affect the color. And the recessive allele allows the persistence of the chlorophyll. It doesn't break down. Okay. So if you take this red pepper, this is what we're talking about. So homozygous for Y and big C, small y, small c. So the y is for the pigment, the c is for the chlorophyll stability. You get the heterozygotes, and you're making the pigment, and the chlorophyll is being stabilized or breaking down. Okay? The dominant allele causes the breakdown. All right? And so when when you got the dominant allele here, this is the color you get. Now you cross those, and you still get the 9 to 3 to 3 to 1 that you would see from a typical Mendelian dihybrid cross, but look at the phenotype. Here you get the red coloration because you have the red pigment and the chlorophyll is being broken down. Here you have the red pigment, but it's not. Here you're not producing the pigment, but the chlorophyll is also breaking down. And here you're not producing the pigment and you're breaking down the chlorophyll. So it's a two-step process. So what you're going to see in almost in all these examples is two genes whose products function in a biochemical pathway. So for instance, this is the pathway for the production of these red and yellow pigments. This enzyme is encoded by the Y gene at the Y locus. This enzyme is encoded at the C locus. Okay. Now, will I give you this figure and ask you to fill in these intermediates? No, hopefully you're going, oh God, I hope not. No, I leave that to the biochemists. They're supposed to torture you with that. Okay. What I want you to understand here is this idea that because the two genes, they independently assort. If they're on different chromosomes, they're going to give you the sort of segregation of 9 to 3 to 3 to 1 that Mendel saw, but it's how the products of those genes interact with each other that affect the outcome. Okay. So the idea is what this slide is trying to tell you is you need to treat the genetics, the segregation of those genes, just as you would with Mendel's rules. Go through the whole dihybrid cross, then look at how the products of those genes interact with each other to give you the different phenotypes. Okay? Any questions? Make sense? Okay. A variation of that is epistasis. This is a concept that a lot of students, I think, will have difficulty with, but it's really pretty straightforward. It's what we just talked about. It's the interaction of the products of two different genes that yield the phenotype. But with epistasis, what we're really saying is because of the particular alleles for one gene, it masks the presence of the other gene. So it's like dominance and recessiveness, except Mendel's dominance and recessiveness only involved one gene. Here we're involving two. I think it'll be easier if I give you an example. But first, just so I have some terms that I can put on the exam, so the epistatic gene is the one that masks the hypostatic gene. All right, so I'll give you an example, and this epistatic gene can either be dominant or recessive. So I'll show you a case of dominant epistasis, recessive epistasis, and double recessive epistasis. All right. And then try to break it all down into a way to think about it rather than just trying to memorize a big table. There's a table in the text, and if you memorize it, you might be able to answer the questions. There's easier ways to do this. Okay. Recessive epistasis. What it means is the presence of homozygous recessive genotype at one locus, at one gene, makes the other alleles, you can't determine what the alleles are at the other locus. All right, so I think it's easier if you see this in an example. An example of this is coat color in Labradors. Really cool dogs, I like them. All right. There's the interaction of two genes. To get the fur color, you have to make the pigment, and you also have to deposit the pigment in the hair shaft. All right, so one gene codes for the enzyme that makes the pigment. The other involves an enzyme that is needed to deposit the pigment into the hair shaft. All right. So now, big B, we'll use B to indicate the gene that codes for the enzyme that makes the black pigment. Recessive allele is brown. Makes a brown pigment. Dominant allele for E, this is the gene that codes for the protein needed to deposit the pigment in the hair shaft. If the recessive allele ends up with yellow hair because it's not functional. With the recessive allele of E, you don't deposit the pigment into the hair shaft. So even if you make the pigment, the hair is still yellow. So again, and the important point, these are two different genes. So if you look at the genotypes that you could get, typical Mendelian genotypes, here you're making the black pigment and you're depositing it in the hair and you have black hair. Here, this is how you get the chocolate labs, because it's a recessive mutation of the enzyme for the pigment production, but you're still depositing it in the hair. However, if it's homozygous recessive for E, you can't tell what these alleles are. You can't tell whether this is homozygous dominant, recessive, homozygous dominant, heterozygous, or recessive, because they all have yellow hair. So E, is that the epistatic gene? E is the epistatic gene, that is the hypostatic gene is B. E is a recessive epistasis, because you only see this effect when E is homozygous recessive. But when it's homozygous recessive for E, it masks or prevents you from determining what the genotype at B is. That's really all epistasis is. One particular genotype prevents you from directly determining what the genotype of the other gene is. Yeah. E is for taking this pigment and depositing it into the hair. So if you can't put the pigment into the hair shaft, it doesn't matter what color pigment you make, the hair will just be a yellow color. Okay? So here's the way you can do the experiment. So you got black labs and yellow labs. You do the cross. You get the heterozygotes. They're all black because you got a dominant allele for both genes. You breed them, and you get the 9:3 to 3:1 ratio that we always see for independently assorting, with two independently assorting genes. But this group, because they're homozygous recessive for E, are both yellow. So you really get a 9:3:4. So what you see is any variation of the 9:3 to 3:1, you're probably looking at epistasis. This is 9:3, but the 4 is really 3 plus 1. These two, instead of having two distinct phenotypes, give you one phenotype. So E is epistatic to B, and big B is basically masking the presence of those alleles. It's recessive because you only see that when E is homozygous recessive. Okay. You can do dominant epistasis. It's the same thing, except the only thing that's going to change are the ratios, because now if the epistatic gene has if the dominant allele is present, then you'll see the epistasis. All right. So here's one that they use from the text: fruit color and some summer squash. So you have a single copy of the epistatic. Okay. So you can take white squash that are homozygous, cross them to green, and the F1 are all white. Now if you intercross those, the F1, you get 12 to 3. You get 12 white squash, 3 yellow, and 1 green out of every 16. Is that a variation of Mendel's ratios? Sure. The 12 is just simply the 9 plus the 3. All right. So it's 3 to 1 for white, yellow, and green. Or you can even look at it as 12 to 4, 3 to 1 for white and some color. That kind of a ratio begins to suggest that you have a dominant allele that's inhibiting pigment production, and that's what leads to the white color. All right. So we can diagram this out. Let's say W is a dominant allele of a gene that interferes with producing the pigment, and you'll have a white color. So if you have the uppercase, the dominant allele of W, you're not going to produce pigment, and you're going to have a white color, regardless of what the other gene codes for. We also see this sort of 3-to-1 ratio between yellow and green, suggesting a second gene involved in color formation. So we'll let Y, uppercase Y, be dominant for yellow to lowercase green. But you're only going to see this locus if, in this case, you're homozygous recessive for a W, because it's a dominant epistasis. All right. So here's the possible genotypes, just like we've been looking at. So here you have a dominant allele for W, which means you're not going to make a pigment, so it's going to be white. So it doesn't matter here what the genotype of Y is. As long as you have this mutated enzyme, you're going to produce white squash. Here, because it's homozygous recessive for W, now you can begin to see the two colors. All right? So allele W is epistatic to Y, and it suppresses the synthesis of the pigment, and it's dominant because a single copy of the big W allele gives you the phenotype. It's a biochemical pathway. So we'll just kind of diagram it out so you can see how it works. So as I said, you see these in these sequential steps in biochemical pathways. So what's happening here is, I'll show you in a diagram, enzyme 1 is going to take a colorless compound, so that would be a white squash, and makes that into a green compound. The second enzyme converts green to yellow. So it's just a simple A to B to C type biochemical pathway. You need enzyme 1. Here, the dominant allele of W codes for a protein that interferes with pigment production. So if you have the genotype, the dominant allele for W, it doesn't matter what else is there. You're going to block this enzyme 1, and you're never going to get past this point, and all of those plants are going to be white. If you're homozygous recessive for W, then you have enzyme 1, makes compound B, which now gives you the green coloration. And then the next step that takes you from green to yellow depends on the genotype of Y. Okay, but again, if you have a dominant allele here, you can't tell what the genotype for Y is because the whole process stops at this point. Yes? I can't think of an example because it wouldn't function as just dominant. Because even the heterozygote here is still going to give you the epistatic effect. Or not. Or I guess unless you're thinking about expanding it out, let's say you had some phenotype that's a result of 3 genes, and 2 could be epistatic, 1 could be dominant, 1 could be recessive. You could do that. Well, that'd make a good extra credit question. Okay. The last one I'll show you, and then kind of try to show you how to think about this more broadly, is duplicate recessive epistasis. All that simply means is, again, we're dealing with 2 genes, and you're going to get the same phenotype whether you're homozygous recessive at the first locus, homozygous recessive at the second, or homozygous recessive at both. All right? So you have individuals at 2 loci are crossed, and you get a 9-to-7 phenotypic ratio. Is that a variation on the 9-to-3 to 3-to-1? Still adds up to 16. All right? So in this case, it looks more like, here's the 7, that the 3 to 3-to-1, that normally have 3 different phenotypes, are all giving you the same phenotype. So the example in the textbook comes from some coloration in snails. So you have 2 different populations of albino snails. All right? And the genotype is, and here's the genotype, you can read them. All right? So they're albinos. You cross them and you get all pigmented snails. When you intercross them, you get 9/16 are pigmented and 7/16 are albino. All right? So you go back, do your dihybrid cross just as you would with Mendel, as we did before. Here's your 9-to-3 to 3-to-1 genotypic ratios. But here, the phenotype is all the same, and so you get 9 pigmented, 7 albino. All right, so how does that work? What you're really going to be doing is 2 steps in the biosynthetic pathway, and if you don't complete both, then you still have the albino. So the presence of the 2 recessive alleles at the first gene, at the second gene, or both, all give you the same phenotype. Probably easier to see it in a figure. All right. So what's going to happen here is, again, we have 2 enzymes, compound A to B to C, but we only get the pigmentation if we successfully are able to convert A to B and B to C. So if we have an epistatic effect, so we have a recessive, homozygous recessive for the A gene, that enzyme, which codes for enzyme 1, blocks us and holds us at compound A. So those animals are albino. But let's say you have a dominant allele. Okay, now it makes compound B, but it's still an albino color. You've got to go through the next step in the pathway, which is encoded by B. If that one's homozygous recessive, then you get blocked here at compound B, and it's still the same phenotype. You have to go through both steps and get all the way to compound C for the pigmentation. So if you fail here, here, or both, you get the same outcome. Yes?
Wouldn't there be an additional situation where you have dominant on both, but there's a problem with production of compound A?
Oh yeah, you could. Oh yeah, you could have that. This is just trying to keep it as simple as possible, not the real world. But yeah, and when we get to the table, there's some other types of epistasis, but we won't worry about those. Okay. Yes. Probably. You could take this same situation, and all you'd have to, let's see, would that work? Yeah, it would reverse the ratio, so the only way, yeah. What would the ratio be? Yeah. Because what would be the only genotype that would give you the pigmented snails? Would be homozygous recessive for A and for B. Okay. So these modified ratios are the results, again, of the gene interaction. So first you want to look at them as, what are the genotypes that would come? They all were based on a standard Mendelian dihybrid cross. So you want to look at the genotypes. And again, it's, as I keep stressing, it's the way the gene products interact with each other. And so the first thing you'll see is the ratios are always in 16ths. Well, the reason for that is you got 2 genes with 2 alleles. All right, so the probability of inheriting any particular combination is 1/16. It's basically 1/4, 1/2 to the 4th power. All right. So what I want to show you next is this table from the text, which has all of this laid out. But instead of memorizing it, take a look at what it's showing you. All right. So up here, the top row is no epistatic interaction. This is the standard Mendelian dihybrid cross with the 9 to 3 to 3 to 1. Well, 9 is when you have at least one copy of the dominant allele for A and B. We'll just use A and B. Doesn't matter what the other ones are. Here you have one copy of the dominant allele for A and your homozygous recessive for B. Here is just the converse. You're homozygous recessive for A, but you have a dominant allele for B. And here you're homozygous recessive for both. All right. When we did recessive epistasis, the coat color in the labs, what you had was 9 to 3 to 4. What it's telling you is, with the part that's shaded in, says these 2 genotypes, which normally would give you 2 different phenotypes, give you the same. So what do they share? They're homozygous recessive for A. So this would be recessive epistasis. All right. But when you see 12 to 3 to 1, what's being shared here? They both have one copy of the dominant A allele. So you got dominant epistasis. Here, you got the 7. What do they all share? they're either recessive, homozygous recessive for A, homozygous recessive for B, or both. There's your double epistasis. Here's the one we just talked about, the dominant, the duplicate dominant epistasis. You get all of these, give you the same phenotype, as long as there's at least one copy of the dominant allele for each gene. The only way you get a different phenotype is to be homozygous recessive for both genes. So if you think about it mechanistically and conceptually, it makes sense rather than trying to memorize what that table shows. Yes?
To go back to the ratio of 9:7, what it said basically, anywhere you
Which one, this one?
Yeah, 9:7.
Right.
Anytime you have a homozygous recessive gene, you always get that ratio.
This says in this particular case, if you're homozygous recessive for B or A or both, they all give the same phenotype. So that's the example where you got to complete two steps in the biosynthetic pathway. If you block it either way, at either point, you get the same phenotype. All right. Mutation of in Drosophila, they have their normal brick red eye color. They found this white-eyed mutant. We talked about why it was pretty easy to recover that because of sex link. And so the gene, the locus that encodes the protein, which is actually, in this case, this protein is actually involved in depositing the pigment in the eye. It's not responsible for making the pigment. But when mutated, it gives the white eye color. It's on the X chromosome. But many different alleles have been found of the white gene. Okay? So when we came out of these experiments, people were doing more experiments, and now they found a fly that has an apricot-colored eye. Somewhere between white, somewhere between red. And the question is this a different gene? And that there may be two different genes that can give you these different eye colors? Or is it an allele of the same gene? So is apricot just another allele of the white gene, or is this a completely different gene that also affects eye color? Everybody understand the question being asked? So you have these two different, okay. So this is where the complementation test comes in. This is to determine, the initial experiments are to determine whether we're looking at two different genes, these two phenotypes, two different eye colors. Are they simply different alleles of the same gene, or are they two completely separate genes that happen to both influence eye color? Okay. So the way you do this test is you take parents who are homozygous for each of the mutations. So you take a white-eyed parent, an apricot-eyed parent, and you do crosses. So the progeny are going to be heterozygotes, right? Okay. If you're really looking at different alleles of the same gene, then the progeny are still going to have a mutant eye color, because neither one is producing the functional protein. But if the mutations are at different loci in different genes, then the heterozygous offspring are going to have a wild-type phenotype. The white-eyed fly is going to be making the wild-type protein for apricot, and the apricot fly is going to be making the wild-type protein for white. When you cross those together, if it was two different genes, they'd be heterozygotes. They now have both functional proteins, and they'd have red eyes. Okay. So complementation occurs if the offspring have the wild-type phenotype. So if you see the wild-type phenotype, you say, yes, we've had genetic complementation. And it tells you these mutations are different genes in a related pathway. Now, I think an easier example, and I think I can probably I think we can do this without rushing. An easier example is, I've got to explain a little bit of genetics about yeast. So yeast is a single-cell fungus. It's a eukaryotic cell. It's going to follow Mendel's rules, hopefully. Okay. One of the nice things about working with it is yeast cells, they have sexes, but they call them alpha, A and alpha rather than male and female. They can be haploid. Everybody know what I mean by haploid? They just have one copy of the genome. So they can be haploid for A or haploid for alpha. If you grow them together, then some of those, an A cell and an alpha cell, the different mating types, can fuse together to form an A alpha diploid cell. Okay. It's actually more fun than that. You can take an A cell, let's say you're growing a colony of haploid A cells. Yeast can undergo something called mating type switching. So some of those A cells can switch to be alpha cells and then mate with themselves. And so you actually end up with a colony of A cells, alpha cells, and A alpha cells. Okay. So if you're thinking about that, you go, why in the world would yeast ever be used for genetics? Well, there's a gene that encodes a restriction, an endonuclease that's needed for this mating type switching, basically sex change. And as long as that's mutated, they can't change. So what we got to think about here is we're going to take a haploid strain. They're going to have a certain phenotype. Mate them with a haploid strain of a similar phenotype, and we're trying to ask, are these mutations in the same gene or in different genes in the same pathway, and we're going to form the diploid. Okay? So here's a simple little pathway. X to Y to Z. We're going to have a gene that encodes enzyme 1. We're going to have a gene that encodes enzyme 2. All right? We have 3 different haploid strains that can't synthesize Z. They're Z autotrophs. So it could be due to a mutation in the gene for E1, or it could be due to a mutation in the gene for E2. So the question is, these 3 strains, we derive them independently. Does Z1, let's say the strain 1 and 2, do they have allelic mutations? That is, they're Z minus because the mutation is in the same, different mutations but within the same gene. Or are they Z minus because they have mutations in different genes but in the same pathway? Now an important part of this to make sense is it's going to be extremely unlikely that if this, let's just say this one is a mutation in the gene for E1, it probably has a functional E2 gene. Okay? And if this happens to be a mutation in the gene for E2, it probably has a functional gene for E1. So I have these 3 different strains. Now when we cross them together, when strain 1 and 2 are crossed, they're still Z minus. But when we cross Z1 and 3, now there's Z plus. In other words, they can make the Z compound in the same way here. So what does that really tell us? When we cross them, and that's what I'm showing you here, is strain 1 and 2 carry mutations, allelic mutations. They have mutations within the same gene, but just different mutations within the same gene. So when we cross strain 1 and 2 together, let's just say arbitrarily strain 1 has a mutation in the gene for E2. That's what I show you with the minus. But it makes E1. When you put the strain 1 and 2 together, they still can't make enzyme E2, and it's still Z minus. But when we cross 1 with 3, and 3 has got a mutation in E1, when you bring them together, now they have both enzymes, and they make Z. So they're put in different complementation groups. So the biochemists did this early on when they were trying to work out the biosynthetic pathways, let's say for making histidine. If I remember correctly, there's about 10 or 11 different enzymes that are needed to make this. So they isolated a whole series of histidine oxytrophs and started mating them together. And if they saw complementation, what did that tell you? That you had mutations in 2 different genes. So these were put in 2 different complementation groups. Then when you cross strains 2 and 3 together, all right, they also complemented. But when you put strains 1 and 2 together, they're in the same complementation group. Eventually, let's say you keep doing this, you make 10 complementation groups, and then every new histidine oxytroph you find will fit into one of those complementation groups. What does that tell you there? That you've probably found all the genes involved in making histidine. Okay? So if you got to do, if they still do these biochemical pathways in biochemistry, all right? And let's say it's histidine, and you would expect it to go, the first gene is his1, the second is his2, his3. but in fact it's like his 4, 6, 7, and that's because they're numbered based on how the order in which they're found, but not necessarily the order in which they function in the pathway. Okay? But this complementation approach was critically important in figuring out how many genes are in this biosynthetic pathway, and then getting a collection of different alleles of those genes, and then the biochemists had to go to work.